So thank you. I'm privileged to speak to you today about what will be my PhD thesis. And what is really exciting to me is the translational aspects of this. So we're excited about data that we're bringing in that's indicative that we'll actually be able to push these to an IND or a filing for a drug that we hope to be able to pull into the clinic. So first, I want to address a conceptual novelty, an advantage that we're now learning about in the field of cancer. So traditionally, cancer has been thought of as a disease that is primarily rooted in genetic mutations or heritable pieces of the genome that have broken down. And sometimes that is addressed in not just heritable genetic breakdown, but also in the breakdown of somatic cells. So as you age, your cells continue to accumulate mutations. And certainly that has a big impact on cancer, but epigenetics, we're learning, is as important, if not more important, in the development of cancer. So this is Waddington's famous epigenetic landscape, and I'm just going to use this to illustrate the basic concept that we are going with. So at the top, you might imagine a zygote or a totipotent cell that then differentiates to a multipotent cell, and then at the end of the day, your body is made up of primarily these terminally differentiated cell types. And cancer is often thought to be a de-differentiated cell type. So it kind of moves back up the pathway and finds or carves out a particular epigenetic thermodynamic minimum at which it sits. And what's interesting is that this doesn't represent necessarily a degradation of the genome at all. So you can form a cancer that has an identical genome, at least in the portions that we're interested in, to a healthy cell. Right? So it's not mutated on the DNA level, but on the epigenetic level, it has had various marks, histone modifications or DNA methylation events that have changed the expression profile of the cancer and caused it to become a tumor or a metastatic burden on a patient. And so what's interesting about this is that if we can push this ball back down the hill, if we can push it to differentiate, we can slow down the growth, make it a healthy cell again. And that is conceptually, you have the advantage of not trying to destroy all of the cells necessarily, but just changing the way that they act. So one of the main success stories so far in this kind of treatment is histone deacetylase inhibitors. And so one of the ways that our cells manage the way that they express genetic information is how the DNA folds or unfolds into either constricted chromatin forms or open chromatin forms. And one of the pieces of the puzzle that determines how your DNA is coiled is the modification on these histone beads that DNA wraps around, and in particular, modifying a lysine tail with an acetyl group by this enzyme, histone acetyltransferase. And of course, this is an equilibrium process with histone deacetylase that removes that mark. And these are known to be major players in controlling the higher level folding of your gene code as it is three-dimensionally sort of folded up into a complex system that hides certain genetic information and exposes others. So it turns out that in cancer, this particular enzyme, histone deacetylase, is very overexpressed and shuts off particular genes. And so by inhibiting that enzyme, we're able to turn back on particular gene pathways that will then reestablish the cell cycle, reestablish the cell's ability to recognize problems and induce apoptosis if necessary. And that's the primary mode of action for this new class of compounds. And this has led to a lot of effort from medicinal chemists who have targeted this enzyme, and they have similarities across the board that are very easy to understand. So in the active site, there's zinc that is used to catalyze the deacetylation of the lysine. And we combine that zinc with the so-called zinc binding group, and the most famous of these is the one on the top, the hydroxamic acid. Then you need a linker to traverse the tunnel, which may be different lengths for different isoforms of the enzyme. And then there's a lot of variability, and this is where it gets interesting. There's a lot of variability in the surface recognition group. You can change up almost anything, and it will still have potent efficacy against these enzymes. So there has obviously been a lot of excitement about this in the clinical world. 500 clinical trials since its inception in the late 90s. And what is startling about this is that many of these trials represent cases in solid tumors. The only two approvals are for drugs that are indicated for cancers of blood. So with all of this effort, all of the money that has been poured into developing these drugs, it's been a failure from its most exciting potential target, and that is breast cancer, prostate cancer, lung cancers, glioblastomas. Solid tumors were the hope, and when it came to phase 2, phase 3 clinical trials, a lot of these drugs fizzled out. And so the big question is why, and how do we address that? So the poor bioavailability and the ineffectiveness in solid tumors is primarily it's a delivery problem. These drugs aren't getting where they need to go. So understanding that and getting around the poor pharmacokinetics of these molecules, we attach to SAW a head group that is effectively it's a homing missile. And this homing missile is based off biolutamide or Casodex, which has made AstraZeneca $580 million just over the last year. So it is very well understood, and these kinds of molecules are well understood to target prostate cancer very well. And a recently approved drug, enzalutamide, that is more similar to our pharmacophore actually, is known to accumulate in the tumor 100 to 200 times more than in blood. So if this pharmacophore can be sucked up by prostate cancer, adding it to the histone deacetylase inhibitor, the hope is at least that that will allow us to overcome the poor pharmacokinetics of the parent compound. So we've done a series of screening. This is a second generation set of molecules where we varied the head group. We first did some experiments where we changed the head group a little bit, but mostly changed the linker length to optimize its binding to both of its targets, right? It's binding the androgen receptor in order to target prostate cancer, but also binding histone deacetylase to actually give it its punch. So against the androgen receptor, our first generation leads didn't get below the 1 micromolar level. But excitingly, our new lead compound is at 32 nanomolar, binding to the androgen receptor, which is an order of magnitude, better than an order of magnitude, improved against Casodex or biolutamide, which is clinically approved anti-androgen. Also, again, this is a second run through. This is kind of a selected example of our best hits so far. Out of these lead compounds, again, our leading compound is hitting at 4 nanomolar for histone deacetylase inhibition, which compared to the clinically approved it's in the same regime as SAHA, which is exciting for us. But what's most exciting is not just its in vitro efficacy against the androgen receptor and histone deacetylase, but what's most interesting to us is that if you look at these molecules against prostate cancers, they're able to do something that neither of the two classes independently could do. So if you look at biolutamide and enzalutamide, which was just approved by the FDA in August of this year, against D145, which is a metastatic version of prostate cancer, it doesn't do anything at all. it's literally like giving buffer to the animals. And of course, they didn't even attempt to give it to patients that had this kind of a phenotype when they went into trials. So, however, even though, so this also, I need to mention, this D145, this metastatic prostate cancer, represents the real problem in the field. So these two drugs are phenomenal for early stage prostate cancers, but for later stage prostate cancers where they become resistant, there is no treatment. There's literally no available efficacious treatment. And most people who get prostate cancer will respond very well to the initial treatments, but then a few years later, if there's any cells that are left, typically 2 to 3 years, they will have this relapse of prostate cancer that is completely untreatable. However, our compounds are still efficacious not only in the hormone-dependent, but also in the hormone-independent versions of prostate cancer. So that is what has us excited about these. And they're also more potent than Zaha, the clinically approved histone deacetylase inhibitor. But what I'm really excited about most of all is this number here. This is in a healthy cell line. And this is in mouse data, which we're getting. But the fact that we don't see any toxicity in a healthy cell line compared to the clinically approved drug, in addition to maintaining the potency against multiple forms of prostate cancer, is what has made this compound truly a lead. Because it's not enough to just be efficacious, you also have to be safe. So far in the drug discovery pipeline, we have made it through the first 2 stages, and we are now embarking in the 3rd stage. So we have done a lot of screening. Of course, we benefit from all of the decades of medicinal chemistry that came before us that we sort of used to even get started. Did our own multiple generations of filtering, and then came up with molecules that are, at least in vitro, very safe and also very potent and selective for multiple prostate cancers. And now we are moving into animals. I didn't show it, but we have our first sets of PK data and ADMET toxicity data back, and we just got that, and we're very excited to see that it is as good as the clinically approved precursors. And so now, in collaboration with a medical doctor at the Medical University of Tennessee, we're doing efficacy studies in animals, and we hope to use that to leverage a larger move forward. So obviously this is a very cost-intensive enterprise, especially as you start to move towards humans. But thankfully, we're covered. We have, this is one patent that is, you can find it online. There's also a couple other ones that are not listed here. And excitedly, the Georgia Research Alliance has funded us now for the second part of phase 1 funding to get this work done and really push these molecules into the clinic. And so it's our hope to acquire SBIR funding and additional Georgia Research Alliance funding, as well as perhaps a strategic partnership to really do the kind of studies we need to do in order to make a true clinical candidate available to patients within the next few years. Thank you.
What questions do we have?
When do you think, when do you start to field studies, and when do you think you'll have some data?
So we've already begun. It's right now, so we're doing PC3 and D145 in mice, and those I expect to have the data back. We've at least told the people who are funding this that we'll have the data back. Our milestones are December for those experiments, and crossing my fingers to see how those go. But of course, if they fail, that's also good because you don't waste a lot of money and more expensive things later on. So,
How do you dose?
okay, so we're doing both oral and IV PK studies, and we're also doing intraperitoneal, I believe, for the first set. And we're doing it with a PC3 and D145 model that has luciferase reporter in it so that we can track it over time so they don't just get endpoint data. But we have to do our MTD studies first, and that's kind of the next step.
So did you say that SAHA is non-specific so SAHA is specific for has no specificity for cancer cells or
I wouldn't say no
cells?
specificity. So the cell line that we tested was a liver cell line. You can also test other healthy cell lines, and it does have the selectivity inherently. SAHA does. Obviously, that's why it's approved.
I guess my question was, why do you want to speculate on why your drug is
Well, because
so
ours
specific?
depends on a number of receptors that are overexpressed in prostate cancer, even the hormone refractory ones. And I don't know if you noticed, but it's kind of a large molecule. So if you don't have the secondary binding effect, at least for our molecule, you're not going to be able to get it into the cell in adequate concentration. I mean, that's the fundamental logic that we use when designing these. So the fact that against liver cells, they are totally not toxic. We're pretty sure it's just because there's the complete absence of the antigen receptor or a couple other receptors that we are also investigating might be targeted.
Do you have evidence, sort of physical evidence that an individual molecule can simultaneously bind to the HDAC?
It's not simultaneous.
No, so
It's
some
not intended to
molecules-
be simultaneous.
So some of the molecules bind to HDAC, some of the molecules will bind to the antigen receptor, and
Correct.
together you have an additive or a synergistic effect.
Yeah, it's a synergistic effect, but we also say that the thing is, you'd expect that it would be, it's going to bind to different things at different points in its lifetime in the cell. Okay, so there are cell surface receptors that bind to testosterone that we believe we've identified with our collaborator, which is exciting because that's been a big controversy in the literature as to what's actually on the surface that binds testosterone. So we know now that our molecules do hit that, and so there, obviously, there's no histone deacetylase enzymes to target, so it's only got one thing. Once it goes into the cytoplasm, now you've got both the antigen receptor and some isoforms of histone deacetylase that are in the cytoplasm, so you have two targets there. And then the idea is that because the antigen receptor is a nuclear receptor that actually translocates to the nucleus, that it would sort of drag our molecule in, and that's where it would have a big effect once it has been released from the antigen receptor in the nucleus, that it could then target all of its histone deacetylases in there.
Any other questions?
How well conserved are the animal model receptors? Are these very well, almost identical?
Yeah, well, we're giving human prostate cancers to the animals, right? So the cancers that they have are from humans, so we wouldn't expect any problems.
Well, that's on target.
Hmm?
That's on target. The rest of the mouse, it's mouse, right? Has the chance of taking in the drug as well if it's got the normal human array of receptors.
Yeah, that's a good point. The receptors that we know that it does target, so for instance, the GPRC6A receptor is very homologous in mice and in humans. So that could become a potential problem, but again, that's not something that we've thought about. So, great question. Is that something that has ended up sort of changing the outcomes when moving from preclinical to clinical in your experience?
I don't have experience in this. I've done the opposite where we've knocked things into mice to change things, but I haven't actually worked in this one.
Thank you.
Well, thank you, Berkley.